Okay, so the last presentation this morning will be given by Rishabh Jain, and Rishabh comes to us from Bemis in Neenah, northern Wisconsin, and but Rishabh is a recent graduate of our university, and in fact it was through some collaborative research with Bemis that Rishabh was introduced to the company and was recruited to work at Bemis. And so we're very happy that that was one of the fruits of the collaboration. And Rishabh received his PhD from my research group in the Department of Chemical and Biological Engineering. And I think he will talk about some of the collaborative research that has gone on between the university and Bemis.
So good morning, ladies and gentlemen. Thanks for the kind introduction. So I am a research scientist at Bemis company, but I graduated recently from the University of Wisconsin under Professor Nick Abbott. And today I'm going to talk about some of the collaborative research that Bemis sponsored at University of Wisconsin. So I'm going to talk about a novel idea that we evaluated and demonstrated, where we used chemoattractants to lure bacteria to contact killing surfaces. So first, a brief overview of contact killing surfaces. So these are surfaces that are decorated with covalently attached cationic molecules. These can be polymers containing quaternary ammonium groups, or they can be cationic macromolecules containing various cations like biguanides, pyridinium, or phosphonium groups. And the way they work is they interact with the negatively charged outer membranes of the bacterial cell membrane. And when the bacteria come in direct contact with these molecules, these displace essential peptides and they increase cell permeability by forming pores in the cell membrane. The advantage with contact killing surfaces, an obvious advantage, is that there is no release of antimicrobial agents. So as a result, although there is reduction of bacteria in the adjacent medium, but there is no increase in toxicity due to the release of these molecules into the medium. However, an inherent disadvantage is also that these surfaces are passive towards bacteria which reside away from the surface. So this is where the idea, we came up with the question that can we attract bacteria towards the contact killing surfaces and thus enhance its antimicrobial efficacy. So we all know, it's widely known that bacteria can be influenced by chemotaxis, or in other words, a concentration gradient of certain chemoattractants can attract bacteria towards the higher concentration of chemoattractant. And so the concept that we wish to demonstrate is shown in the figure on the right. So a conventional contact killing surface, which would be represented in A, would have microbes away from the surface, and only those microbes which are in the vicinity would be killed. However, if we could establish a gradient of a chemoattractant near the contact killing surface, what we wished to show was that we could attract the bacteria and towards the surface and kill them. So to demonstrate this idea, we modified a widely used assay which is used to quantify chemotaxis. So this is known as the capillary assay. So the figure on the right shows, it's a simple assay, but it's pretty versatile. What you have is a microcapillary containing a few microliters of a chemoattractant of interest, and this is immersed in a bacterial suspension. And what happens after this is that the chemoattractant will diffuse out of the capillary, forming a concentration gradient around its mouth, and this will attract the bacteria towards the capillary mouth and into it. And after a certain period of time, you can count the number of bacteria that have entered the capillary and thus measure how effective the chemoattractant is at attracting that particular strain of bacteria. What we did is we modified this assay slightly by coating the inner walls of this capillary with a contact killing antimicrobial, which was DMOAP. And now we looked at 2 different bacterial strains and 2 different chemoattractants. And what we wished to show now was, number one, can we attract bacteria into these capillaries and then do these antimicrobial surfaces, are they effective at killing them? So this slide just shows the contact killing surfaces that we fabricated. So glass was coated with the antimicrobial DMOAP, and this XPS spectra here shows evidence of the C/N ratio being 26, showing that there was a multi-layer DMOAP coating formed on the glass surfaces that we used. And to evaluate the efficacy, we put these microcapillaries in bacterial suspensions, and what this graph shows here on the right is that the contact killing antimicrobial coated on the surfaces was highly effective at killing the bacteria that was put inside it relative to controls, and over a period of 90 minutes it would be able to kill 99.99% of the bacteria inside. So now I'll show a video of what happens near the capillary mouth when there is no chemoattractant present. And so in this video, you will see that I have selected a number of bacteria and shown their trajectories in order to show more clearly how bacteria move around the capillary when there is no chemoattractant present. And so 60 bacteria were randomly selected around the capillary mouth, and what you find is, number one, that the bacteria are moving randomly, and while a few of them move towards the capillary mouth, most of them are moving in an unbiased fashion in various directions. So this is the control. Now, what happens when we put in a chemoattractant inside this capillary? So how does that influence the movement of bacteria? And again, in a similar fashion, what you see is now that the bacteria are moving in a much more directed fashion, a lot of them towards the capillary mouth, and quite a few of them even enter the capillary mouth. So this is just to establish that the assays that we conducted, bacteria was indeed entering the capillary mouth. And we looked at these bacterial trajectories near the capillary mouths for various cases. So one in which there was no chemoattractant present in the capillary. In B, you have bacterial trajectories for microcapillaries that contained a chemoattractant, and in C, you have the trajectories for microcapillaries that contained the chemoattractant as well as the contact killing antimicrobial on its surface. So the essential thing to notice, so two things. Number one, that the bacterial movement is dominated by runs over tumbles in presence of the chemoattractant. So as a result, you see that the bacterial trajectories are much more defined and directed towards the capillary mouth. And then secondly, bacteria is drawn into both untreated and the contact killing chemical treated microcapillary. And so this bar chart just quantifies the number of bacteria that we counted that entered the microcapillary. And what you see is that when there is no chemoattractant present, there are very few bacteria which enter it. However, when there is chemoattractant present, 8 to 10 times that of the control number of bacteria enter the capillary mouth. And this is irrespective of whether the microcapillary was treated with the antimicrobial or not. And so what essentially I've been trying to show here is that first, can we get the bacteria into the capillary? And now we come to the next stage, is to evaluate if the contact killing antimicrobial is able to kill the bacteria that we have attracted towards this system. So to do that, the next step was for us to is to take out those microcapillaries and count the number of bacteria that are present inside them. And this is a bar chart that I will explain further. What it shows is that you have the control where you have no chemoattractant present in the microcapillary or in the bulk. So let's look at this image on the bottom right. So this is our system, this is the microcapillary, and you can, and we have established a chemoattractant concentration gradient if this capillary contains chemoattractant and which attracts bacteria inside it. So for our control, we have a system where there is no chemoattractant present on either side. And in that case, we find that the number of bacteria in the microcapillary are very few in number. Now, another control is if we have the chemoattractant present uniformly inside the capillary and outside that of the capillary. So there is no concentration gradient. In that case, also we see that the number of bacteria is few in number, although it's a bit higher than the first case. And that is just because the chemoattractant provides some energy to the bacteria which move a lot more, which are more motile, and therefore increase in numbers in the capillary. Now, coming to the case where we established the concentration gradient outside the capillary. In that case, we see that we have attracted a lot of bacteria, around 8 to 10-fold over our controls inside the macrocapillary. And to this system, when we add the contact-killing antimicrobial, we show that we have reduced the number of bacteria. So we are, in essence, killing all the bacteria that we attracted inside the microcapillary. These results are for the system where we looked at E. coli as our model bacteria and aspartate as our model chemoattractant. We also showed these results with another strain of bacteria and other chemoattractants. So in these cases, we see that glucose is the chemoattractant, whereas E. coli is the bacteria, and we also looked at salmonella, which is a common food pathogen, along with aspartate. And so the trend remained consistent, and what we are able to show that across different species of bacteria and 2 different chemoattractants, we are able to replicate the phenomena. So to conclude, the contact killing efficacy of a surface can be enhanced by up to 20-fold due to a nearby concentration gradient of chemoattractants. And so this was a proof of concept to show in general a principle that we can perhaps extend the efficacy of a contact killing surface to beyond its vicinity into a surrounding medium. And so these results were general across 2 common gram-negative food pathogens, and we used 2 different chemoattractants. And so this concept now could be useful in a range of applications in the future. So for instance, surfaces near food source, for instance, packaging of liquid or semi-solid substances. A concern would be that you would be able to kill, with just contact killing surfaces, you would be able to kill microbes that are just near the surface. But what if you could draw the bacteria away from the bulk of the food into the packaging surface and thus kill a lot more microbes? So with that, I conclude the presentation. Thanks. I think it is possible to get to those kinds of levels. I think the numbers that we got are limited by the model system we used. And so the microcapillary that we used has very few microliters. It has very limited space. So you could only attract a certain number of bacteria. But if we broaden this principle to a bigger surface, then I believe that we could actually amplify the effect a lot more.
So are there gaseous chemoattractants known for bacteria? Gas phase.
Yeah. So the question is, are there gaseous chemoattractants available for bacteria? Known gaseous chemoattractants? Yeah, not that I know of, but yeah, I don't know of any gaseous chemoattractants. But if anybody in the audience knows, please feel free to share.
If they do exist, and they may be things like CO2.
Yeah.
Then one could think about in the context of food packaging, using gas phase concentration gradients to draw organisms to cells.
Yeah, and I know of phototaxis, which is light influencing movement of plants and maybe even cells, and perhaps, I don't know, maybe oxygen or some gas which might be able to attract bacteria towards it. So, but that's an idea we could look at.
And I think you can also use chemorepellent to keep the bacteria away from surfaces, right? So rather than attracting them, you can just not come closer to the
Just to repeat the comment.
Yeah, and so the comment is, what if we use chemorepellents and that would repel bacteria away from a surface? I guess one may think of maybe two surfaces where one is repelling the bacteria towards another surface, which is contact killing. So,
or like maybe you can use these surfaces for catheters in urinary tract infections. So bacteria usually swarm through these catheters and infect your urinary tract. If you have these chemorepellent surfaces, the bacteria cannot crawl through your catheters to the urinary tract, and you can use them in healthcare applications.
yeah, and yeah, that's a good idea, I guess, where you don't want bacteria to enter geometry at all. You might want to actually repel them by controlled release of chemorepellents.